This lesson will explain the structure and function of F-pumps, also known as ATP synthesizing complexes or ATP synthases. These elegant molecular machines produce nearly all of the cell's ATP. They are located in the inner mitochondrial membrane, where they serve as the final components of oxidative phosphorylation. Major constituents are labeled. Most of these will be defined during the course of this lesson. The energy comes from the inner mitochondrial membrane, where it is stored as a hydrogen ion gradient. The gradient is produced by oxidative phosphorylation. Hydrogen ions flow through the FO domain down their electrochemical gradient. The F1 domain uses energy from the hydrogen current to drive ATP phosphorylation. The FO domain is the transmembrane component of the complex. The A subunit contains two channels. Hydrogens load at channel 1. Hydrogens pass through channel 1 to a C subunit binding site. The C subunits form a turbine that shuttles hydrogen around to channel 2. Channel 2 releases the hydrogen ions. As the hydrogen current flows through FO, it turns the turbine. The F1 domain projects into the mitochondrial matrix. F1 has 3 alpha and 3 beta subunits. It is the beta subunits that have the ATP synthase activity. Energy from the turbine is transduced through the gamma subunit to induce conformational shifts of the beta subunits. The beta in the foreground begins in the open conformation. In the open conformation, beta binds an ADP and phosphate. The loose conformation secures ADP and phosphate binding. Tight has an even higher binding affinity for ADP and phosphate. ATP synthase acts during the tight phase. It drives ADP phosphorylation, producing ATP and water. When beta returns to the open conformation, it releases the ATP and water before it binds the next ADP and phosphate. The basal view of the F1 domain displays sequential ATP synthesis with all 3 beta subunits. From the basal view, the cycle proceeds clockwise. Gamma acts as a rotor. As it turns with the turbine, it induces conformational shifts of the beta subunit. As gamma turns, the F1 domain pumps out ATP. Thus, metabolic energy from the hydrogen current is transferred to ATP, the primary energy currency of the cell. This has been an excerpt from the Biology from Molecules to Embryos educational program. Check it out.